Introduce our group members, Safira, Nabila and Jin. This video is about Practical 2A, titled Spectrophotometry, Sugar Analysis using the 3,5-Dinitrosalicylic acid DNS method. The objective of this experiment are to introduce the student another example to the use of spectrophotometry for preparing glucose standard curve. Second, to determine the absorbance of each test tube contain a different volume of glucose solution. Third, to plot the standard graph of the absorbance y-axis against the concentration, which is x-axis. And the last objective is to determine the concentration of reducing sugars in each test tube. These are the materials used in the experiment: spectrophotometer, glass beaker, hot plate, cuvette test tube rack, micropipette and tips, distilled water, standard glucose solution, Rochelle site, and DNS region. Experimental procedure. The first step, all reagents including distilled water, glucose solution and DNS region are pipetted in duplicate into a series of dry clean and the test tube are labeled 1 to 6. The pipette must be held in 10 degree to 45 degree against the wall of the receiving chamber to allow the liquid to flow smoothly down and to prevent contamination or inaccurate volume. Next, the test tube are shaken well to ensure the reactant to be uniformly mixed for the reaction to proceed efficiently. Next, the test tube are boiled in hot water bath for 5 to 7 minutes. This step will help to increase the rate of reaction between the DNS region and reducing sugar. Then, after the test tube being cooled, 1 ml of Rochelle salt are added to impart the stability to the color of the product form. This stabilization is crucial for precise and reliable absorbance reading. Next, the solution is transferred into the cuvettes. During the process, inspect cuvettes for scratches, cracks, or air bubbles, as this can distort absorbance readings. Make sure to not shake or tilt the cuvette roughly after filling to avoid creating bubbles. Last step, the reading of absorbance are tested and recorded using spectrophotometry tools. The reading starts from test tube 1 to test tube 2, test tube 3, test tube 4, test tube 5 and test tube 6. Of this experiment, based on this picture, the color pattern gradually changing from light to dark from test tube 1 to test tube 6. Then the difference in color occurs due to the varying concentration of reducing sugar present in each sample. The higher glucose concentration, the darker color of the solution. The result of the absorbance. Based on this result, the absorbance of test tube 2 is the lowest due to the lowest concentration of glucose solution in test tube 2. Meanwhile, test tube 6 contains the highest concentration of glucose solution, thus the absorbance is the highest. This is the graph of glucose solution versus absorbance. The linear equation obtained is y equal to 1.3573x plus 0.0252. The plotted graph shows the positive linear relationship between absorbance and the glucose concentration, which is consistent with Beer-Lambert law. When glucose concentration increases, the absorbance also increases. Based on the graph plotted, the concentration of reducing sugars in each test tube can be calculated using the linear equation. The highest content of reducing sugar is belong to test tube 6, while test tube 2 have the lowest content of reducing sugar. The initial glucose solution concentration influences the concentration of reducing sugar calculated. The higher initial glucose concentration, the higher concentration of reducing sugars. In conclusion, the experiment was successfully done and all the objectives are successfully achieved. A Another example to the use of spectrophotometry for preparing glucose standard curve can be introduced to the student. Spectrophotometry measures absorbance to create a standard calibration curve using non-reducing sugar concentration. After that, the absorbance of each test tube containing a different volume of glucose solution can be determined by spectrophotometry tools. Test tube 2 has the lowest absorbance while the highest absorbance belongs to test tube 6. The third achievement is the standard graph of the absorbance against the concentration can be plotted by using the result obtained. The graph shows a positive linear relationship between absorbance and the glucose concentration. The last one, the concentration of reducing sugar in each test tube can be determined based on the linear equation from the graph. Thus, test tube 6 contains the highest amount of reducing sugar while test tube 2 the lowest. Then for our group reflection, we learn new method to determine the amount of reducing sugar by the NS method, and then we can observe how the NS reagent react with reducing sugar to form a color compound which increase with the concentration. Next, there were several challenges we faced during the experiment. The first one, air bubble in the pipette tips can lead to inaccurate volume transfer and data. And the second one, ensuring constant timing throughout the heating process, as prolonged or insufficient heating can cause the reaction color intensity to change rapidly. So for the suggestion for improvement, for the pipette problem just now, when aspirating, make sure the tip is completely submerged and pipette slowly and steadily. For the timing problem, heating and absorbance measurement timing should be standardized to guarantee consistent result.